The goal of the presentation is really to come at the end with five or six ideas about what should be the next clinical trials in the field of genomics. So let's start with the current status of precision medicine in metastatic breast cancer. So what we are doing now in 2016 is that we are developing drugs, in genomic segment, meaning we are evaluating drugs in patients with genomic alteration. So this is the summary of where we are in terms of drug development according to genomic alteration. So here the title is Recurrent Driver Alteration in Breast Cancer, and the key word here is recurrent. So we are focusing on genomic alteration that are targetable and that are recurrent, meaning they occur, let's say, in more than 1% of patients, meaning that you can constitute cohorts of group of patients who share the same genomic alteration. In breast cancer, we have 6 genomic alterations that are targetable and for which there is some evidence of drug activity. So the first ones are BRCA1, BRCA2 mutation. I will not discuss germline versus somatic, just introduction. So that provides some sensitivity to PARP inhibitors. We have AKT1 activating hotspot mutation. This is 4% of breast cancer, and that is associated with high sensitivity to AKT inhibitor. HER2 mutation associated with sensitivity to neratinib. We have mutation, and this is the most important in breast cancer. We have around 25% of breast cancer who present mutation on the alpha isoform of PI3 kinase. So again, these are activating mutation, hotspot mutation, on a gene whose name is PIK3CA, and that codes for the alpha isoform of PI3 kinase. And early clinical trials suggest that if you give an alpha-specific PI3K inhibitor, there are some objective response. And finally, we have some mutation on ESR1. So again, activating mutation, hotspot mutation, in patients who became resistant to endocrine therapy, and this is associated with response to estrogen receptor degrader. So these are the 6 targetable genomic alterations in breast cancer. Maybe there are more, but until now, 2016, these are 6. Maybe we will reach 10. It's unlikely we go above 15. Some waterfall plot. So this is the activity of neratinib in HER2 mutant breast cancer. You can see some objective response. Again, efficacy of AKT inhibitor in patients with AKT1 activating mutation. A patient who presents breast cancer are in pink, so you can see some objective response. And finally, waterfall plot here for patients who present PIK3CA mutation and who are treated with alpha-specific PI3 kinase inhibitor. So these are illustrations from phase 1, phase 2 trial that indeed, when you give a targeted therapy matched to one of the 6 genomic alterations, you have some objective response. So this is a starting point. We have 5 to 6 recurrent targetable genomic alteration that have been associated with objective response. Interestingly, and we can discuss that later, you can see that most of them are hotspot mutation on oncogene. Until now in breast cancer, with the exception of BRCA1, that is kind of a particular gene, we don't have any high sensitivity associated with mutation on tumor suppressor gene. Hotspot mutation, oncogene. What are the next challenge? And I will not go into detail, it's just to mention them. Here we are speaking about challenges in drug development. First, what is the clinical impact of having multiple genomic alteration in the same tumor. I'm not speaking about two cancer cells presenting two different alterations. I'm speaking about multiple driver alterations in the same cancer cell. So first question, is it associated with primary resistance? And there's some data from Encyclopedia Cell Line that it could be the case. Okay. Second question, how to develop drugs in rare genomic segment. We have seen that some of the genomic alteration occur in 2%, 4% of breast cancer. And here the challenge is how can we screen large number of patients in order to get the 200 who present the genomic alteration. Challenge number 3, what is the optimal tool to generate a genomic portrait of metastatic disease? And here the question behind this is whether the use of circulating DNA could be interesting to screen patients who present genomic alteration, because circulating DNA can be done in patients who have bone-only disease, that is a little bit more difficult if you do biopsy, and can be done in centers who do not have the interventional radiology facility to make biopsy of metastatic sites. So the question of whether we can select patients based on circulating DNA to develop drugs in genomic segment is really now a hot one. Okay? So let's move now to some questions that are really related to clinical research in genomics for metastatic breast cancer. The first question is it useful to sequence a large panel of genes? We had some discussion during the break and even before. So this is what we are doing now. We stratify a frequent disease into 5 or 6 smaller group of patients, and we develop drug in each group. Okay? What we know, and this is extremely important because this is the switch from 5 to 15 genes to more than 100 genes. And what we know is that conventional drug development is being done in patients who present targetable recurrent alteration, meaning genomic alteration who can see in more than 1%. Okay? What we see when we use large panel of genes or when we do whole exome sequencing in a retrospective cohort is that, in fact, you have a large number of patients who present with a targetable genomic alteration that is almost unique to this patient. Okay? Example, in breast cancer, activating mTOR mutation. It's very unusual. I've seen twice in my life. So we cannot make trial testing mTOR inhibitor in this patient because it's only in 1 or 2 patients. Okay? So we have large number of patients when we do sequencing who present a targetable genomic alteration that is almost unique. And this is why we should maybe offer large panel of genes in patients. Okay, and that explains the switch from few number of genes with validated genes, recurrent, to large number of genes. Okay. Do we have evidence that this is clinically useful? So what we have now, we have reported data from patients, a case report in the literature, that if you screen gene alteration that are very rare, you can have some objective response. Example, this case from Institut Curie. So I come from the center with a neighbor to Curie Institute, so usually we compete. But this case is so interesting that I can cite them. So this is very, very nice case. And they are very strong in biology. So this is a case, HER3 mutation, HER3 activating mutation. It's very, very, very rare in breast cancer. And believe me, it's very unusual. They have identified this case, mutation in different sites, okay, and they gave dual HER2 inhibitor, and they got objective response. Okay, so this is a typical case for which you have done sequencing of genes that are mutated in a very rare number of cases. So you have offered large panel of genes to cover some very rare gene, and you get an objective response. Okay, so for this patient, it was 100% useful to run large panel of genes. Okay, but the question, especially in France and other countries, is that okay, this patient is good, but is it useful for the population of patients? It has been shown to be useful for some cases. Is it useful to the population of patients? If we want to address this question, theoretically, we have to run this kind of trial, comparing what we discussed before, 5 to 10 genes, who will be the standard of care in the next 3-4 years. And we have to compare this to the use of large panel of gene, meaning more than 100. But large panel of gene is not enough. What you need, and actually this is the value, is the method to interpret the genome. Because sequencing 100 genes, I would say it's what I understood from my colleagues of biologists is not difficult. But being able to interpret 400 genes and to pick up what is the genomic alteration that must be targeted, actually that is the challenge. Okay? Still, if we want to move to the large panel of gene, we would like to have some data comparing large panel versus small panel. Okay? What are the data now? Until now, there are some trials who try to address this question. In the literature, we have at least 6 trials. Until now, these trials, randomized or not, could not provide evidence that it is useful to run large panel of genes. Okay? Why? And there are 2 explanations for that, and this is extremely important. We were one of these trials. We have run this trial. Well, it was kind of pilot study, but at the end, only a few number of patients could get the targeted therapy, and few of them had objective response. So overall, when you run this kind of large panel of gene trial, the results now do not provide level 1 evidence that it is useful. Why? There are 2 reasons for that. And this is now well identified. The first reason is that these trials testing large panel of genes, did not match the right drug with the right genomic alteration, and this is extremely important. We know now that these trials were not doing the right matching. Example, in some trials, investigators matched mTOR inhibitor to mutation on PI3 kinase. But we know now that this is not the correct matching. The efficacy of mTOR inhibitor is not predicted by mutation on PI3 kinase. So most of this trial, in most of the patients, did not perform the right matching. So this is why these trials are not interpretable, basically. The second reason that is more prospective is because the method to identify the driver in each patient, was not optimal. Basically, that was discussion between biologists, bioinformaticians, and clinicians who believe they know what is the biology, but the reality is that at the time these trials were done, people were not capable of interpreting genomics because we are missing a lot of information in terms of biology of PI3 kinase, MAP kinase, etc. Okay, so the key point here is that at the time where these trials were done, validated and robust tools to interpret biology in individuals were not available. Okay, so people believe they know, but in fact they didn't know. And I was part of them, so I can criticize easily. Okay, so these are the 2 reasons why these trials testing large panel of genes fail. Okay, it does not mean we should not do it. It means that if we run this trial, we have to match the drug well, and we need new tools to interpret genomic data. So let's go now. It's going to be shorter, the next part. So to other possible applications of genomics in patients with metastatic breast cancer. So first possible application is to identify patients who could be sensitive to immunotherapeutics. So it has been reported in several studies now in melanoma and lung cancer that the more there are non-synonymous mutations and the more there are neoantigen and the more patients are sensitive to immunotherapeutics. Okay? Interestingly, in metastatic breast cancer, there is one population of patients that you can see here. One population of patients who are around 10% of patients with hormone receptor positive breast cancer who present very high mutational load. Okay? And this is the starting point to start clinical trials testing immunotherapeutics according to the number of mutations. And so, for example, we started a clinical trials using whole exome sequencing to know the number of mutations per patient and give them immunotherapeutics. So this is one possible illustration in 2016 about the possible use of whole exome sequencing in clinical trial. The whole exome sequencing, mutational load, and then we drive patients to immunotherapeutics in clinical trials when they have high mutational load. Okay? Interestingly, this patient with high mutational load, they also have mutation on PI3 kinase. And that could be the starting point of combination between immunotherapeutics and PI3 kinase inhibitors. Okay? So where we are now, until now we have discussed a baseline. Okay? We have discussed whether we can identify the driver and whether we can identify patients who are eligible for immunotherapeutics. So we treat the patient when we have the drug, because basically this is the problem, having the drug. So when we have the drug, we treat the patient. Okay? If we were right, patients have objective response, residual disease, and then in some patients, a secondary resistance occurs. Okay? And the next question is, how can we use genomic tests, okay, to better understand and characterize the secondary resistance? So this is the field clearly of circulating DNA. And there are some reports now in breast cancer that circulating DNA, cell-free DNA, can detect relapse or resistance before imaging. We have an example in this case from the Royal Marsden Hospital, where you have a rise in circulating DNA without metastatic relapse, and 6 months after, or maybe a little bit less, you have this metastasis that occurs. So circulating DNA could be a useful tool for the early detection of resistance and can detect a subclonal alteration involved in resistance. Okay? So here the clinical research question will be whether the early detection and targeting of subclonal alteration involved in resistance is useful. In other words, what kind of clinical trial we need here is the comparison between patient monitored by circulating DNA to detect the resistant clone and treat it versus a group of patients for which we will not monitor circulating DNA and we will treat them at the relapse. Okay, so this kind of trial now is being designed and developed, and hopefully in the future we will be able to show that early detection of resistance using circulating DNA can improve the outcome because it allows early targeting of the resistance. And there are some data starting to emerge in lung cancer about that. In breast cancer, the obvious indication will be ESR1 mutation that mediate resistance to endocrine therapy, and PTEN mutation who mediate resistance to PI3 kinase inhibitor. Okay, so last 2 points, because I know we are short on time, so I'm stressed about the timing. So last 2 points. First, can we use genomics to stratify patients based on outcome? So usually when you say that, people say, prognostic test, we don't really care. In fact, this is not true. So why to identify genomic segment associated with very poor outcome? So now there is a research field that emerged that consists in identifying which genomic segment predicts very poor outcome in metastatic breast cancer patients. So people say, oh, we don't care about that because we treat them anyway. This is totally wrong. Because if you can identify a group of patients based on genomic test that present a very poor outcome, that allows to have fast-track drug approval based on phase 2 data. So what I mean here is that if you show that a targetable genomic alteration is associated with less than 1 year overall survival, and you show that the targeted therapy has 1 year PFS in the phase 2, you will get approval based on the phase 2 data. At the opposite, if you are unable to show that the genomic segment has poor outcome, you will have to run phase 3 because metastatic breast cancer is starting to be a little bit more chronic disease and is difficult to get approval based on phase 2. So this research area is extremely important. And for example, these are very preliminary data just to illustrate. ERCL1 mutation has been associated with poor outcome in this study that we did in France, but also now in one study in US and one study from UK. So maybe it's going to be a genomic segment associated with very poor outcome where some drugs could be developed very quickly. Same for mutational load. The more there are mutations and the more prognosis is poor. This is based on a cutoff at 150 mutation, but this prognostic value is also significant when the mutational load is considered as a continuous variable. So probably it is quite a robust indicator of outcome. Okay. Last point, we discussed a lot about DNA, but in fact there is also some room and some development for RNA and protein assay. Why to use RNA and why to use protein assay? Basically, at least in breast cancer, is to identify which pathway is activated. Example, in one study, the identification of patients with mTOR activation here with phospho-S6 was associated with a better sensitivity to mTOR inhibitor. This is just one illustration that we have DNA for the driver, immunotherapeutics, resistance, but on the top of that, it will be probably useful to add RNA expression and even some phosphoprotein assay. For which drug? For endocrine therapy, anti-androgen, mTOR inhibitor, and CDK4 inhibitor. Okay. So conclusion. So what we have now? So what we have now is a model where we test drug in cohort of patients who share the same genomic alteration. And for this, we have 6 genomic alterations now in breast cancer. It can go to 10 or 15, but it will not go to 50 or 100. When we are speaking about recurrent genomic alteration with drug development. The next question here is to run clinical trials testing the clinical utility of large panel of genes. But for this, we need robust method of interpretation. Otherwise, we just do nothing. Future trial should test whether the early detection of clonal evolution is useful, and this is really the field of circulating DNA. Okay. Maybe one opportunity for immunotherapeutics in estrogen receptor positive breast cancer, because some of them present high mutational load, and we need tools to quantify pathway activation because this is really complementary to DNA. Detecting a mutation is good, but if you can say that the protein is activated, I mean, that is a good complement to DNA. Okay, I will not discuss the last point because I do not have slide for this presentation about challenges in terms of implementation of precision medicine, and this is different model in each country, so it's a nightmare for a global company, for example. It must be very difficult, but okay, that's it. And thank you for my colleagues and the funder for all these things. So, voilà.